This is my ASM virtual lecture entitled How Life Changes Itself, the Read-Write Genome. I'm Jim Shapiro from the University of Chicago. And what I'd like to talk about is how we think about genomes and how they change and how the cell uses them. The conventional wisdom is that the genome tells the cell what to do and that it's a read-only memory. And the only way the genome changes is through copying errors. I've included a quote here from Sidney Brenner, a Nobel laureate from the 2012 Alan Turing Memorial, where he talks about the genome as a read-only memory in a von Neumann machine, which can construct copies of itself. And the only source of heritable variation is by copying errors. Actually, we've learned a great deal from studying the process of genome change. And I'd like to give you a different view, which is that the genome is a read-write memory system. And in order to help you understand where I'm going with my lecture and why I'm including certain examples, let me give you the conclusions at the start, and then we'll come back to these at the end, which is where they belong properly. So the only constant in life is change at all time scales. Cells change as they go through the cell cycle. External conditions change, organisms undergo change, and cells undergo change during multicellular morphogenesis. Cells read their genomes dynamically to adapt to changing conditions during cell reproduction and multicellular development. And the ways that cells read their genome is a function of prior inscriptions in the genome: coding data, signals for interaction with cell molecules, nucleoprotein complexes, chromatin formatting, and so forth. And these are forms of genome inscription, which is an active physiological process at all biological timescales. And in the course of evolution, cells actively write data and formatting signals into their genomes, and they do this by a variety of active processes, including symbiogenesis, horizontal DNA transfer, and what I like to call natural genetic engineering, or cells cutting and splicing and rearranging their DNA. Now, life operates at least 3 different time scales, and it's useful to keep these in mind when we think about how the cell makes inscriptions on its genome. Within the cell cycle is one time scale, and various events like metabolism, DNA replication, and so forth go on during the cell cycle. And so that's one biological timescale. A second is over multiple cell cycles, as for example in multicellular development, or the growth of a vegetative population of an organism that divides mitotically. Finally, there's evolutionary time, and this includes all events which lead to taxonomic divergence and the origination of novel characters. And as we'll see, evolutionary time scales can be quite short as well as quite long. And the major point of what I'm trying to say is that cell physiology, cell biochemistry, makes active genome inscriptions at all 3 time scales. Within the cell cycle, the way the genome reads and maintains it's read their genomes, and the way they maintain their genomes requires that there have been prior inscriptions into the genome. This is like formatting a computer program. So what kind of information does the genome contain? Naturally, it contains coding sequences for RNA and protein structure, but it also contains signals for transcription, signals for processing RNA once it's been transcribed, signals for translating the RNA into protein, signals that regulate DNA replication so that it occurs once and only once during each cell cycle, signals that influence chromatin formatting for long-term memory, signals for spatially organizing the genome within the cell, and we're learning a great deal recently about how important genome formatting is, genome function, both in terms of expression and recombination, and replication and transmission. And finally, signals that help the cell repair damage to the genome when that damage occurs. Now, there are different classes of genome inscription at the three different timescales that I've mentioned. So during the cell cycle, there are transient nucleoprotein complexes that are formed. And these are complexes that form and then come apart. And examples include replication and transcription initiation complexes, repair complexes, and the very important cell cycle checkpoints. And these complexes assemble and then disassemble quite rapidly so that they provide a short-term picture of the status of the genome, but they're only used for a very short period as necessary during the cell cycle. Over multiple cell cycles, we have another form of genome inscription, which we call epigenetic inscription, and we now know that that is involved with chromatin modifications. And epigenetic modifications are inherited during cell divisions. They are stable over multiple cell cycles. They play an important role in multicellular development. And then chromatin changes can be imprinted and subject to rewriting across generations. And we're beginning to learn a great deal about transgenerational modifications of chromatin inscriptions in response to various environmental and behavioral inputs. Finally, there are inscriptions at the level of DNA content, DNA sequence and structural changes to the genome. These, of course, are heritable over multiple cell generations and over evolutionary time. They arise because of various processes, symbiogenetic fusions of cells, horizontal transfers between cells, virus infections, and a whole variety of processes that I like to include under the title Natural Genetic Engineering. Now, to exemplify what I mean about the formation of nucleoprotein complexes within the cell cycle, on the left here we see a picture of lac repressor protein bound to two different lac operator sequences making a loop in the DNA, and below the crystallography picture there's a cartoon of what's happening, and this is important in the short-term metabolic regulation. Whether the cell utilizes lactose or not is controlled within the cell cycle by this complex. And this complex contains information about the history of the cell, the availability of glucose, the availability of lactose, and this is a short-term metabolic regulatory nucleoprotein complex. On the right is shown the DNA damage response in yeast, and it shows how the yeast continually monitors its DNA, and when it detects damage, it activates both repair processes and checkpoint processes, which send signals to the cell cycle apparatus to halt progress through the cell cycle until the DNA damage has been repaired. Without such cell cycle checkpoints, cells would be formed which have damaged genomes or incomplete genomes, and this would be injurious to the population as a whole. Checkpoints are extremely important processes in the completion and the accurate completion of the incredibly complex process of the cell cycle. Over multiple cell cycles, genome writing occurs by epigenetic modification, And that's illustrated here in this cartoon, which I borrowed from the Scientist magazine. We now understand epigenetic inscriptions in the genome as including DNA methylations. They're shown here in the upper right. The cartoon is a little bit incorrect in that it indicates the methylations on the phosphodiester backbone, whereas in fact they occur on the bases, on adenine in bacteria, and on cytosine in both prokaryotes and eukaryotes. Those are the sites of methylation. In addition to the methylation of the genome, there is the complexing of the genome into nucleosomes, which are illustrated on the left here. And the nucleosomes form these very compact structures where the DNA is wrapped around histone molecules. And the interesting thing we've learned about epigenetic formatting is that the histones have tails. They have amino-terminal tails which stick out from the nucleosome. And as illustrated on the bottom, these various amino acids in these tails are subject to chemical modifications. They can be methylated or acetylated or hydroxylated or phosphorylated. And the cell can read these modifications to know what kind of chromatin the DNA is encased in and how to operate on that chromatin. So some chromatin is suitable for transcription and replication. This is called open or expressed chromatin. Other chromatin is silenced or heterochromatic and is not subject to transcription. These changes can be, chromatin formatting can be maintained over large distances within the genome and over many generations. So, for example, during cellular differentiation and multicellular development, large regions of the genome can be turned off because they're not needed in certain specialized cells and tissues. Now, all of the things that affect the organism can result in reprogramming the genome. Here's an example of two different chromatin formatting events at the agouti locus in mice. And the difference between these two mice is not what happened to the mice themselves, but how their grandmothers were fed. So this is transgenerational, multi-generational transmission of a chromatin formatting difference, which leads to lack of expression in the large pale mouse on the left, or active expression in the small dark mouse on the lower right. Over evolutionary time, genome writing occurs by multiple processes, and these include cell fusions. And what's illustrated here is a phylogeny of all of the eukaryotes, and it indicates the symbiogenetic origins of the eukaryotic cell, as well as its photosynthetic descendants. All eukaryotic cells carry a mitochondrion or a defective descendant of a mitochondrion. Those are labeled H and M, which stands for hydrogenosome and mitosome. Those are defective mitochondria. And the fact that the mitochondria is an indwelling descendant of an alpha proteobacterium tells us, and has its own unique ribosomes and DNA replication apparatus, tells us that at the origin of the eukaryotic cell, there was at least one symbiogenetic event where a bacterial cell, an alpha proteobacterium, came to be an endogenous component of the larger eukaryotic cell, which had a different DNA replication, transcription, and translation apparatus. Later on in the history of eukaryotes, a second bacterium became an endosymbiotic organelle. That's the cyanobacterial ancestor of the chloroplast or plastids that's present in all photosynthetic cells. And this gave rise to the lineages which led to green algae and red algae. and green plants as indicated on the bottom of the figure. But what's also indicated is that there have been multiple secondary symbiogenetic events leading to the formation of new photosynthetic lineages, because red algae or green algae have become engulfed and endosymbiotic within other groups of eukaryotes which are not normally photosynthetic. So Euglena, for example, has gone into a taxon of eukaryotic cells, which also includes the trypanosomes. And in one of these secondary symbiogenetic cells, there are now 4 different DNA compartments. There's the nucleus, there's the mitochondrion, and then there's the algal plastid-derived compartment, and then another compartment called the nucleomorph, which is the former nucleus compartment of the algae. And DNA transfer occurs from these secondary compartments to the nucleus, and probably in the reverse direction as well, although that has not been as well studied. So over time, the mitochondrial and plastid genomes become deprived of coding sequences. They migrate to the nucleus, and then the proteins are made cytoplasmically and imported into the organelles. So this is a major form of genome rewriting by cells fusing, and as we can see, it's not a rare event. It's happened multiple times in eukaryotic evolution. And there are also cases of endosymbiogenesis in prokaryotes as well. Now, over evolutionary time, we know from the study of antibiotic resistance in bacteria, for example, that genomes can be rewritten by horizontal DNA transfer and by cells acquiring DNA from other cells. What's illustrated here on the left are the 3 mechanisms that we know about for DNA transfer between cells. Cells can take up naked DNA from the environment by a process called transformation. Cells can transfer DNA from one cell to the other by a process called conjugation. And these 2 processes involve the action of DNA transport pores in the envelope of the cells, and they're actually very similar transport pores used in uptake of DNA from the environment, transformation, and in conjugation. A third way that DNA is transferred from cell to cell is by enclosure in a virus particle, which can originate in one cell and carry DNA from that cell to a different kind of cell that it infects. And this is actually very common in nature, and it's called the process of transduction. And what is shown on the right is the results of these horizontal transfers, which are the introduction of various mechanisms for inactivating antibiotics, changing the targets of the antibiotics so they're no longer sensitive, or even creating pumps to remove the antibiotics from the bacterial cell. It's interesting to note that in the 1960s and 50s and 60s, there was a conventional theory of antibiotic resistance which depended upon spontaneous mutation and modification of bacterial cell structure. This theory was consistent and could be confirmed by laboratory experiments. It was possible to isolate sequences of mutants that had high levels of antibiotic resistance. But when scientists went out into nature and asked how was antibiotic resistance spreading in the clinics once we were using antibiotics in medicine and agriculture, it turns out that horizontal transfer and various other mechanisms like plasmids and transposable elements and so forth were important components of the antibiotic resistance evolution. And the bacteria were using mechanisms that were completely unknown to conventional evolutionary thinking. Now, horizontal transfer is not just does not just occur between prokaryotic cells in the bacteria and also in the archaea and between them. It occurs in eukaryotic cells. And this is shown here in an example of plant parasitic nematode worms, which need to digest the plant material that they're going to use as their food source. And rather than evolving their own hydrolytic enzymes for digesting plant polymers, they've acquired them from bacteria or from fungi. And what's shown here is a taxonomy of plant parasitic nematode worms. And what's interesting is that different taxa or different groups have acquired different hydrolytic enzymes that they need from bacteria, from different bacteria and different fungi. So there have been multiple horizontal DNA transfers into these nematode worms. And we know that animals and plants, both of them, can acquire DNA sequences from microorganisms and indeed from each other as well. After horizontal transfer, the transferred DNA has to be integrated into the genome. And living cells possess a variety of biochemical tools for altering their genomes. I call this the natural genetic engineering toolbox. So as we've seen, they contain pores for DNA mobilization across the membranes of the cell. They can take up DNA or transfer it from cell to cell, so this is important for horizontal transfer. But cells contain nucleases and ligases for cutting and splicing DNA. They contain polymerases for replicating DNA, for making new DNA, for correcting it, for proofreading it. And they also include specific error-prone mutator polymerases, which can introduce genetic changes, localized point mutations, into the DNA. And it turns out that a large percentage, perhaps the vast majority, of small mutations in the DNA are due to these error-prone polymerases. They contain excisionases, which can remove incorrect or damaged bases from the DNA, such as uracil. They contain proteins called helicases for unwinding double helices and removing damaged oligonucleotides from the DNA. They contain annealing proteins, which are needed in recombination, such as the REC-A protein. They have site-specific recombinases, which combine cutting and splicing of the DNA at specific signals in the DNA. They have reverse transcriptases for converting RNA into DNA. And then finally, they have proteins called transposases and integrases, which are involved in cutting and splicing specific structures of DNA and integrating them into the genome. The transposases and integrases work on mobile genetic elements, which fall basically into three different classes, as illustrated in this slide. There are DNA transposons, which have defined ends. There's segments of DNA which can be cut out of the DNA by transposase activity and then inserted into a new site by the same transposase proteins which remain bound to the ends. And these DNA transposons are then mobile cassettes which carry information from one place to another. There are also retrotransposons which move through RNA intermediates, and they come in two varieties, those which have terminal repeats like retroviruses and go through a complex structured complex process of reverse transcription to form a transposon-like cDNA, double-stranded cDNA, with integrase proteins that are analogous to the transposases so that they can integrate at a new site. And then there are other kinds of retrotransposons called lines or long interspersed nucleotide elements and signs or short interspersed nucleotide elements, which are transcribed into RNA. And then the RNA is used copied into DNA at a cleavage site in anywhere in the genome, and the target DNA serves to prime reverse transcription. This is called target prime retrotransposition. And this process is very nonspecific for the RNA, which is integrated into the genome and can apply to line RNA and move line elements from one place to another. It can move sign elements using the line element encoded proteins, the polymerase and the RNA binding proteins and the nuclease. It can also mobilize messenger RNA and other processed RNAs into the genome to create pseudogenes and other repeat structures in the genome. So this is quite a versatile process, this target primed reverse transcription. These elements have been extremely important in evolution. For example, here's a figure from the first draft human genome sequence, and we can see that in our own evolution, there are over 3 million of these mobile genetic elements, transposons, retrovirus-like elements, and lines and signs. And this means that in our evolutionary history, there have been at least 3 million different integration events of mobile genetic elements. And that's, they comprise, at this time, 44% of the genome, which is about 30 times more than the protein coding DNA, which is about 1.5% of the human genome. Now, over evolutionary time, these mobile elements can help write new DNA sequences encoding new protein domains, which is very important for protein evolution over the course of life history. And what's illustrated here is what happens when a mobile element, in this case a line L1 retrotransposon, inserts inside an exon, and how sequences within that element can be exonized to form extended new coding sequences for polypeptides that will be novel protein domains. So in the B, for example, in A we see a genetic locus which is transcribed and then spliced and has 4 exons encoding the protein, various parts of the protein. If an L1 element inserts inside that third intron, we see that after insertion, various kinds of changes can occur, and 2 of them are illustrated here. One is that new transcription can start inside the L1, continue up to a splice donor site, and then that splice donor site can recognize the splice acceptor site at the 4th exon of the original locus. So a region of the L1 that is transcribed will become the 1st exon of a new protein, and then the 4th exon of the original protein will become exon 2 of the novel protein. So that's one way that a sequence from an L1 element can become an exon encoding a protein domain. And what we see in C is that these inserted mobile elements contain both splice donor and splice acceptor sites, so that a segment of the inserted mobile element can become an exon, which is now included as a 5th exon in a new spliced messenger RNA, and thereby give rise to a new protein coding sequence. So one of the problems of originating novel protein coding regions is solved by this process of exonization, and this replaces the idea of having to change proteins 1 amino acid at a time over incalculably long geologic or biological timescales. Here, a new protein coding sequence can come into being within a single generation. Now, when mobile elements insert next to a functioning genetic locus, one of the other things that they can do is modify the expression of that locus and its regulation. And as we know from evo-devo studies, altering the expression of various genetic loci is often an important adaptive change, even more important than changing the sequence of the encoded protein. So inserted mobile elements can provide new signals for epigenetic regulation, and indeed many epigenetically imprinted loci contain mobile elements segments as the imprinted element, which allows the epigenetic modification. They can come in as cis-regulatory elements for transcription, such as novel promoters or novel enhancers. They can also become promoters and lead to transcription in the opposite direction and produce antisense transcripts, which can play a role in preventing messenger RNA expression. The inserted elements can also become targets for microRNAs and for heterochromatin formation targeted by the microRNA. And then finally, we know that many non-coding RNAs, cis or transacting non-coding RNAs, originate from mobile element insertions. And so they can both act as targets for these RNAs and also as sources for new non-coding RNAs to play regulatory functions. Repetitive DNA and mobile elements also play a role in maintaining telomeres, in forming centromeres, and as has been mentioned, in forming heterochromatic regions. So they do a great deal in terms of formatting the genome and mobilizing signals from one place to another. One of the evolutionary mysteries that's solved by the existence of mobile genetic elements is how are distributed networks of coordinately regulated functions created in the genome. To do this by single nucleotide or accidental changes at each genetic locus, one at a time, would take an impossibly long period of time. But the movement of mobile genetic elements can rapidly transfer common recognition signals to many places in the genome. And that's illustrated here in the figure on the left, where it's shown that the same element at different locations in the genome can respond to a common DNA binding protein. And on the lower right is simply a reference which shows that transposon-mediated rewiring of gene regulatory networks contributed to the evolution of pregnancy and placental development in mammals. So we know that these networks have been created and formed using the ability of mobile elements to go to new locations in the genome and thereby distribute common signals to multiple sites. Natural genetic engineering, like all biochemical processes, is subject to regulation. And we know that there's a wide variety of stimuli, both biological stimuli and non-biological stimuli, which can activate natural genetic engineering, which normally is kept in check by cell regulatory functions. And among the most important of these stimuli are starvation, infection, changes in ploidy DNA content, and hybridization when two different species or two different populations mate with each other and they disrupt the normal control processes which keep the natural genetic engineering functions, in particular mobile genetic elements, under control. And then they become active and create clusters, rapidly create clusters of genetic changes. Mobile genetic elements can be targeted within the genome by standard processes. Here is a figure illustrating how protein-protein interactions target the insertion of retrotransposons in yeast cells to different locations. At the top left is an element called TY3, which is targeted just upstream of tRNA sequences by interaction with two of its integrase protein with Pol3 RNA polymerase III transcription factors. The TY1 element also inserts upstream of tRNA sequences, but at a different position, and there the integrase protein interacts with nucleosomes that are specific for RNA polymerase III transcription. In fish and yeast, the TF1 element inserts upstream of Pol2 transcribed sequences, and here the integrase protein interacts with a transcriptional activator protein to localize it upstream of the Pol2 transcriptions. The TY5 element in budding yeast is very interesting in that it localizes to silent chromatin, and it does that because the integrase protein contains a targeting domain that is phosphorylated and normally interacts with the SIR4 protein that's part of the silent chromatin domain. But if the cell is stressed, the phosphorylation is lost, and now the TY5 can integrate into many other regions of the genome under stress conditions when genetic change may be helpful. Over evolutionary time, genome inscriptions have been extremely important. Here's one paper from 2011, which looked at the human genome in light of 29 sequenced mammalian genomes and found over 280,000 conserved mobile genetic elements that were common to that derived from mobile genetic elements. And about 19% of all conserved differences between marsupials and eutherian or placental mammals could be traced to the activity of mobile genetic elements. More recently, the reference at the bottom points out that in primates, the majority of primate-specific regulatory sequences are derived from transposable elements. So the ability of these elements to move from place to place and carry transcription and other regulatory sequences with them is of great importance has been of great importance in evolutionary history. So we return to our conclusions. The only constant in life is change at all time scales. To deal with change, cells read their genomes dynamically, and that allows them to adapt to changing conditions both during cell reproduction and multicellular development. And genome reading is a function of prior inscriptions, which, as we have seen, is a function of prior natural genetic engineering processes, which have incorporated coding data, signals for interaction with cell molecules and transcription and replication, the formation of nucleoprotein complexes, and chromatin formatting. Genome inscription is an active physiological process at all biological timescales. It is subject to regulation, to being activated and inactivated, to being targeted within the genome, and we have yet to explore how extensive that regulation is. And finally, we know that in evolutionary history, cells have actively written data and formatting signals into their genomes by the processes of symbiogenesis, horizontal transfer, and natural genetic engineering. Thank you.